Welcome life science learners to another lesson. Trust that you guys are well and excited about what we're going to do today. In our lesson today, we're going to focus on revising meiosis, a very important topic. It's important that you remember that meiosis is in both paper 1 and paper 2, and hence it's crucial that you understand the process well and that you're able to revise this section and work out different types of questions. In our lesson today, we're going to have an overview of the process of meiosis. We're going to try and understand the unique characteristics of each phase so that in a test or in an exam, when you present it with the diagram, and as we would work on later, you would be able to identify these stages. So, as we do in any revision lesson or when you're revising, it's important that you look at some of those important terms and concepts that are fundamental to remembering any section. And I always say that in life sciences, having a list of terms is important as you revise a section. So let's look at some important concepts as we get into the overview of the lesson, and then we will try and look at the specific processes and the different stages of meiosis. So let's get straight into it, guys. So as I mentioned, important terms. We'll review the main concepts around the overview of meiosis, and then we'll discuss some application-based questions as we get into the lesson. So, as I mentioned, terms. It's important that we draw reference to the process of mitosis, something that you've done in grade 10. Mitosis is often a process that we understand in terms of how cells produce copies of themselves. When we complete meiosis in grade 12, we often look at a comparison between the two processes. And that's again to help you understand and differentiate between what meiosis is and what mitosis is. So we've got to recognize that meiosis is a type of division that produces gametes, and it's involved in two important processes in humans. We refer to them as gametogenesis, where spermatogenesis and oogenesis produce the gametes. And so we recognize that meiosis is fundamental to producing gametes. The concept of a somatic cell and how these somatic cells are cells that have a full set of chromosomes, and the context of gametes being cells that have half the full set of chromosomes, or we refer to them as cells that are haploid. The concept of haploid and diploid again refers to cells and their chromosome number. When we talk of somatic cells, guys, we often refer to cells that are diploid or having a full set of chromosomes. Remember that full set refers to a set coming from the egg and a set coming from the sperm, and that collectively gives you a 2n complement or a full set. So refer to that being diploid. We also recognize that your gametes are cells that are haploid, having half the full set, so these would be having N and N. So we often refer to the gametes as having half the full set of chromosomes. Let's move on to some more terminology. The concept of polyploidy again refers to cells that have multiple sets of chromosomes, and we look at the advantages of polyploidy in commercial food crop production and the concept around how polyploidy happens, and we see the advantages of that to the production of food and the quality of food crop. We also look at the concept of aneuploidy, where we see there's an abnormality in the chromosome number, and often we will reference the process of how non-disjunction, or when something goes wrong during meiosis, where we have the non-separation of chromosomes resulting in a gamete that has either an extra or one less chromosome, which then eventually fertilizes to form an organism that has a chromosome abnormality in terms of the number of chromosomes. So either one less or one more would be aneuploid. The concept of being able to identify what a chromosome is, and often I use this image to use that comparison. So here we have what we refer to as a homologous pair of chromosomes. Again, chromosomes that are similar in shape, size, and have genes at the same location, so homologous, and that these contain chromatids, and so these are the structures that are produced, that are held together by the centromere, an important concept in identifying the structure of a chromosome, and that centrioles are produced during cell division, where they produce the spindle fibers. So again, we will look at those structures in the process. The concept of homologous chromosomes, as I've just mentioned, chromosomes that are similar in shape, size, and having genes at the same locus, and locus is the position of a chromosome of a gene on a chromosome. Right, the concept of karyotyping and how karyotypes are used to look at chromosomal abnormalities or the number of chromosomes, and to determine the gender of an individual. The Human Genome Project, and what does the genome of an organism mean? So it's the full set of all the genetic information in an individual cell that has been sequenced, and that often refers to the genome of an organism. The concept of a bivalent and how that forms during what we refer to a process called crossing over, when pairs of chromosomes come together and exchange genetic information between inner chromatids, and the concept of what that point of crossing over is called as chiasmata, and then finally the importance of crossing over, which we discuss later. It's important that we look at independent assortment and chromosomal mutations as important concepts in either producing variation in organisms and mutation in individuals. Cytokinesis refers to the division of the cytoplasm, and karyokinesis, the division of the nuclear material that happens during meiosis and mitosis. And then finally, the relevance of meiosis to gamete production, so the processes of spermatogenesis and oogenesis as being two important processes in understanding gamete formation. Okay, it's important that we look at an overview of the process, and we recognize that meiosis consists of two phases, meiosis 1 and meiosis 2. We often recognize that by looking at different phases in meiosis 1, and we know that there's one interphase that happens just before meiosis 1, which is then followed by prophase 1, metaphase 1, anaphase 1, and then telophase 1. It's crucial that we remember the acronym PMAT, and also that we know that in meiosis there's always a number attached to any stage. It's either prophase 1 or prophase 2, and that's again in relation to whether it's the first meiotic division and the second meiotic division. So if you write any phase without a number, and if you're indicating prophase, that again is the context of mitosis. And it's important that you recognize that prophase in meiosis is either prophase 1 or prophase 2. And so never write a phase without a number when we refer to meiosis. All right. And so we do recognize that meiosis 2, or the second part of meiosis, contains 4 phases, which are prophase 2, metaphase 2, anaphase 2, and telophase 2. And guys, as we wrap this segment up, this is an overview of the process. I'd like to spend some time getting into the overview of each process and then some questions. But before we get into that, you guys deserve a little break, stretch break, refreshing, and I'll see you in a bit. Welcome back, guys, from the little break that you had. Trust that you're refreshed and excited about the next segment. In this segment, we're going to spend some time overviewing the different phases of meiosis. It's important that when you revise this section, that you have an image or a diagram that has these processes. What I essentially do in this segment is look at each phase and describe the key characteristics that will help us identify how to identify processes and phases when we're looking at it. Let's get straight into the lesson and try and attempt to look at an overview of the process. So as I mentioned, when we look at meiosis, it occurs in 2 different phases. And I'm going to make sure that I've selected the right color so that it makes it easier for you to be able to see. So during meiosis 1, we find that the homologous chromosomes separate. And that's key to identifying why the chromosome numbers reduce. When we see that, we start off with interphase, where we see that the chromosomes undergo a process called DNA replication, and we find the doubling of that genetic information. We've looked at DNA replication in the past. This is then followed by prophase 1, where a very important process occurs, and we often refer to that as crossing over, where homologous chromosomes lie next to each other in a bivalent, and here you can clearly see that undergoing what we refer to as crossing over, where the inner chromatids exchange genetic information. And these create recombinant copies, and that is important to producing variation in the gametes. From prophase 1, we move on to metaphase 1, and guys often use the acronym META, meaning meet. And so these guys now meet along an imaginary equator as pairs, and we find that now we've arranged these chromosomes along an imaginary equator. We can clearly see that the spindle fibers are now holding the centromeres of these chromosomes in position. From metaphase, we move on to anaphase 1, and this is where we see the separation of the chromosomes. And here we see that the chromosomes now are pulled apart to the opposite poles by the contraction of these spindle fibers. So as these spindle fibers contract, we find that the pair of chromosomes separate and are pulled apart. And that is fundamental to how the pair of chromosomes now move to the opposite sides. And finally, we find that in telophase 1, these chromosomes have moved towards the opposite ends of the cell. We find that the nuclear membrane starts to reappear, and we notice that the cells start to separate out by a process called cytokinesis, where the cytoplasm starts to pinch and produce two separate cells, and we often see a cell plate being formed or the invagination or the movement of the cytoplasm apart. And that's a wrap of the first meiotic division. And again, this cell would have been a diploid cell. We can see that it's got 1, 2, 3, 4 chromosomes, and at the end, these cells will each have 2 chromosomes each. And we refer to this as a phase in which there is a reduction in the chromosome number. And that is clearly seen because of anaphase. It's also important that we recognize that variation happens in this process. And variation happens in 2 significant places. It happens here during crossing over and during the independent assortment of these chromosomes when they move apart randomly. So again, these are important processes which create variation in the gametes at the end. Guys, now when we look at the second phase of meiosis, or meiosis 2, it's important that we recognize that during this phase, sister chromatids separate. Now it's important that we recognize when chromosomes move apart, and we saw that during anaphase 1, but in meiosis 2 we find that at some point we find that the chromatids are pulled apart, or in this case, the concept of sister chromatids being pulled apart. So let's start with, at the end of telophase 1, we find that there's a phase of interkinesis, so between meiosis 1 and meiosis 2, where we move into prophase 2. Again, in prophase 2, we see that the centrioles split, the nuclear membrane disappears, spindle fibers form. Essentially, it's important that we know that crossing over only occurs once in prophase 1. Prophase 2 is simply a process in which the cell now prepares for the rest of prophase of meiosis 2. Right, so we see that towards metaphase 2, we find that the centrioles have produced spindle fibers, and these spindle fibers attach to the centromeres of the chromosomes. We recognize that the chromosomes now meet at the equator. They're no longer in pairs, and so they align themselves in a single line where each chromosome has its pair in the opposite cell or in the other cell. So the pair for this is in that cell, and so they've moved apart in separated cells, which we saw here during anaphase 1. It's important during anaphase 2 that we recognize that this is where the chromosomes have split and moved apart as chromatids to the opposite poles. And then finally, at the end, we see cytokinesis separating out the cells to form 2 cells that each have an n number of chromosomes. And that is where we find telophase 2 occurring, where the nuclear membrane reappears, and we have 4 cells that are now each haploid or having half the full set of chromosomes compared to the cell that we started off in interphase. So that's an overview of the process. Now let's get to some questions and see how we can attempt to answer these questions. Right, so it's important that we recognize that during meiosis that there's a reduction in the chromosome number. I'd like to spend some time looking at that before we attempt that question that I promised to work with. So, prophase 1, when we look at the characteristics of that, we know that the chromosomes condense. We see that the nuclear membrane starts to disintegrate. So we're seeing that in prophase 1, the nuclear membrane, patches of that start to disintegrate. It's important that we understand that the homologous chromosomes now start forming bivalents. And essentially, a bivalent is when chromosomes lie next to each other in pairs. And so we often refer to pairs of chromosomes, but a bivalent is when those pairs come and align themselves along with each other. So we find that bivalents form, and during bivalence we know that a process of crossing over occurs, and that crossing over is the first reason where variation occurs during meiosis, and we refer to that as at the point called the chiasmata. We then look at metaphase, and as I mentioned, the concept of meta meaning meet, and during metaphase 1 we find that these pairs of chromosomes now meet along the equator, and the spindle fibers from the opposite centrosomes will connect the bivalents at their centromeres and align them along the middle of that cell, which we refer to as an imaginary equator. If we go on to anaphase 1, we know that these spindle fibers start to contract, and we see that the chromosomes are pulled apart, which we see distinctly happening in anaphase 1. And then finally in telophase 1, as I mentioned, we see the process of cytokinesis occurring, where the cytoplasm separates out. We also know now that the chromosomes have moved to the opposite poles, they've condensed, the nuclear membrane has reformed, and we have finally 2 daughter cells which are haploid, again meaning having half the full set of chromosomes that they originally were in this cell. So if this was 2n, we end up with 2 cells having half the chromosome number, meaning haploid. Right. When we look at meiosis 2, again, I often use this acronym to say, well, meiosis 2 is PMAT, but it's a reminder of what happens in mitosis. So if you remember what happened in mitosis in grade 10, this is essentially what happens in meiosis 2. It is a type of mitotic division where the 2 cells divide to form 2 cells each having the same chromosome number. And it's interesting, here we see that the chromosomes now split up into its sister chromatids, which essentially are copies of the genetic information. Let's see and recognize how this occurs. Right. So as I mentioned, in prophase 2, the chromosomes condense, which we saw in prophase 1. The nuclear membrane dissolves, again similar to prophase 1. However, there is no crossing over, guys. That only happens in prophase 1. So the cell simply prepares for the next phase, and we find that they move to the opposite poles, which produce the spindle fibers as you see in this image here. These spindle fibers then, sorry, let me go right here. That's where I should be referencing that. That's prophase 2. And these spindle fibers start forming from the opposite poles during prophase 1. We clearly see the nuclear membrane disintegrating in those white areas there. During metaphase 2, as I mentioned, these chromosomes now meet along this imaginary equator with their spindle fibers attached to the centromere. And that holds them in position in preparation for the chromatids being separated during the next phase. As we move on, we find that it's important that we now see how anaphase 2 takes place. In anaphase 2, guys, we know that this is when the centriole, let me just illustrate exactly what happens. So this is when the centromere splits up, and we find that the chromatids are pulled to the opposite sides. And so that's clearly what is happening here. During metaphase, they met, and now we're seeing that the centromeres are pulled apart, causing the daughter chromatids to move in the opposite direction. So spindle fibers contract, they separate out the sister chromatids or daughter chromatids. The chromatids then are now called chromosomes, move to the opposite poles. Guys, this is where we need to pause, and we need to ensure that we recognize what is a chromatid. Guys, when a chromosome is illustrated as that, where you've got the chromatids attached together, it is still a chromosome having 2 chromatids. However, it's important that we recognize that these chromatids are copies of each other. So essentially, when they separate out, it's the same information that has just moved apart. So we still keep the same genetic information, except that we're moving them to the opposite sides. And that is why essentially we find that each cell at the end has the same number of chromosomes that we started off with. So if we have N here, these will still have N number of chromosomes in each of them. And finally, towards the end of meiosis 2, we find telophase 2. And telophase 2 again refers to the chromosomes condensing, becoming more compact, the nuclear membrane reappearing around them, and the cells, the cytoplasm dividing by cytokinesis to form 4 individual cells, each having half the full set of chromosomes. So that is an overview of how meiosis takes place. The final outcome of meiosis is the production of 4 haploid daughter cells. It's important that we recognize that the 4 haploid daughter cells are all unique and have their own variation. And that explains why you and I are very different from our siblings. And I often remind the learners to think about animals that have multiple offsprings at one time. So if you think of dogs and if you think of cats, when they have several kittens or pups, each of them are unique. And that's because there are multiple gametes being fertilized, and each gamete is unique. And that is a good reminder of why in meiosis many gametes are produced and each gamete is unique. So these cells may be genetically distinct if crossing over occurs in prophase 1, which causes the recombination of sister chromatids. We need to recognize that variation during meiosis takes place in the following phases. Prophase 1, where we refer to as crossing over, again during anaphase 1, where we see independent assortment. And what does that mean? That again refers to how these chromosomes have arranged themselves and have assorted. Assorted means separate. We do need to recognize that assortment is independent. What that means is that if you look at this pair of chromosome, the green moves to the one side and the purple moves to the other. Or maternal, paternal separate out independently from the other pairs. When we look at this pair, the purple moved to the one side and the green to the opposite side. So each pair of chromosomes separates out independent from the other, and that combination creates more variation in the gametes that are produced. So what we recognize is that variation happens during prophase 1. We saw that it happens during anaphase 1 as well as during anaphase 2, where each of those chromatids, which are unique now because of crossing over that has happened there, also are pulled apart independently. And this creates significant opportunity for variation in the gametes. And that explains the basis of why individuals show such significant variation. Welcome back, life science learners, to our revision lesson. We continue to revise meiosis. We've looked at some terms. We've looked at the important concepts. Now let's try and apply our understanding to a few questions. Question 1. The diagrams below represent different phases of meiosis in an organism. It's important that when you have a diagram that you read the heading. The heading often gives you a description of what is happening. And here in this diagram, it tells you different phases of meiosis. Let's recognize the fact that in meiosis, we have different phases, and these may not necessarily be in the correct sequence. Right, so let's move into that. So here we're finding there are 3 diagrams that have been given to you, each with a unique representation of what is happening in these cells. Let's read further into the question. The first part of the question requires us to identify A. Guys, when we look at the diagram, we can clearly see structure A being labeled, and structure A points to this little structure. Guys, we know that chromosomes have chromatids that are held together by what we refer to as the centromere. So structure A is pointing to that protein that holds the chromatids together. After replication has taken place and copies of DNA have been wrapped around histone proteins to form condensed structures, they are held together by these structures which are called centromeres. So structure A is the centromere, and it's important that you're able to recognize that. Let's look at part B, or identify what structure B is. If you look at structure B, guys, it points to a bracket that encloses or all of those two chromosomes together. When we look at a bracket that collectively wants you to label two structures, the context of that would be that it's pointing to a pair of chromosomes. The next bit would be, are they similar in shape and size? Looking at that, yes, they are. So for me, that would be a pair of chromosomes. Because they are similar in shape and size, I often refer to them, and the correct term would be homologous. So B would be a pair of homologous pair, and that's a homologous pair of chromosomes. And so that tells us that you've recognized that they are similar and that there is a pair of them. Structure C. Guys, when we look at structure C, and those are structures that are attached to the centromeres, and that structure are those structures are the spindle fibers that are produced from the centrioles during cell division, and they essentially control the movement of the chromatids or the chromosomes in the cytoplasm, in the nucleoplasm during cell division. Right. So as I mentioned, spindle fibers or spindle threads that you can use to answer them. Question 1.2. Identify the phases represented in diagram 3. So guys, when we look at diagram 3, it's important that we first recognize, what does it show? Is it showing you chromosomes or chromatids? And again, it's important that when we look at diagram 3, we can see that there are chromatids indicated. The next question that you've got to ask yourself, are these chromatids? Where are they? So it's important that we recognize that during meiosis 2, that chromosomes are moving apart. Sorry, let me say that again. It's important that we recognize during meiosis 2 that the chromatids are going to be pulled apart. And here we're seeing that the chromatids are being pulled apart. And so clearly this is meiosis 2, and the reason would be that chromatids are being pulled apart, and that rationale would help us to identify that it's anaphase 2. So that diagram there indicates a phase where the chromatids are being pulled apart, and that is in anaphase 2. It's important that you always remember that there is always a number at the end of a phase when we're referring to meiosis. With the exception of interphase, all other phases, prophase would either be prophase 1 or 2. If there is no number, then you're referencing mitosis. So remember that there has to be a number when we're talking about prophase, metaphase, anaphase, and telophase. Right. As I mentioned, that was anaphase 2. Let's move on to 1.3. Write down the numbers of the diagrams to show the sequence in which the phases occur. Now guys, here you're seeing 3 different cells, probably in a sequence, not necessarily showing all the cells in the sequence. So if we were to arrange these in the correct sequence, we would have to understand what is happening in each cell to be able to chronologically place these in the correct order. So when we look at diagram 1, we can see that diagram 1 for me shows you the formation of spindle fibers and centriole splitting. We can see that there are a pair of chromosomes. I often recognize this as part of prophase 1, where chromosomes are now arranged and the spindle fibers forming. So this would be definitely prophase 1, and I'm going to use the acronym P1. Again, that's not what you're allowed to write in your papers. You've got to say that it is prophase 1. Okay? Diagram 2 shows you the meeting of the chromosomes at the equator, and so this would be metaphase 1. And how I know that it's metaphase 1 is, firstly, metaphase is at the equator, and 1 because we see that there are pairs of chromosomes meeting at the equator. And this is going to be anaphase 2, and here we can clearly see that chromatids are being pulled apart. So these diagrams illustrate essentially the process in the following sequence. So we would start with 2, 1, and 3. So again, this would be the initial process of prophase 1, this would be prophase 2, and then finally here you've got anaphase 2. So as I mentioned, here you can see that it's prophase 2, and purely because you've got a single chromatid for each pair, here you've got 2 of them, so that takes us to prophase 1. So that would be, the correct order would be 2, 3, and 1. The next question would be, state one difference between metaphase 1 and metaphase 2. Guys, during metaphase 1, it's important that we recognize that we still have pairs of chromosomes. So when these meet along the equator during metaphase 1, we find that a pair of chromosomes meets at the equator. So that's a pair of chromosomes, maternal and paternal pairs at the equator. And that would be metaphase 1. However, in metaphase 2, we need to recognize that we no more have pairs of chromosomes. So we have single chromosomes that are meeting along the equator. And so the difference between metaphase 1 is that there's a pair of chromosomes that have met along the equator, whereas in metaphase 2, you now have single chromosomes, one from each of the pair arranged along the equator. So in a human cell, you would have 23 of these aligned along the equator in metaphase 2. However, in metaphase 1, you will have 23 pairs of them aligned along the equator. And that's the significant difference between metaphase 1 and 2. Right, so as I mentioned, that's the explanation to that question. Guys, the next question talks about the following diagrams. The diagrams below represent different phases of meiosis. Again, these questions often require that you're able to identify unique phases in this process. It's important that you look at the phases and you identify features in each one of them. As we wrap this segment up, I'm going to assist you in identifying the following parts, and let's get into it. So identify part A. And if we look at the diagram, part A again points to this structure here, and that structure again produces the spindle fibers. So I challenge you to go and reflect on what are those structures that are producing the spindle fibers. I'll hold on for a bit until you give me the answer, or think about that. The next one is structure B. Again, structure B points to a bracket that encloses all of that, and clearly that points to a structure which is separated out from its partner or pair, and that would be one single chromosome. And finally, structure C. If you look at structure C, it points to the cell, and it points to the outer cell membrane, and here we find that's the nuclear membrane here, but the outer wall is the cell wall or cell membrane in that case. So as I mentioned, that could be, A could be the centriole or centrosome, B would be the chromosome, and C could be either the cell membrane or the plasma membrane, depending on the type of cell that we see. So guys, that's a wrap for this segment. Trust that you've been able to apply some of the basic identification traits of each phase. Let's get back after the break and tackle some more interesting questions. See you in a bit, have a quick break, and I'll catch you at the end of that. Welcome back, life science learners. Let's have a look at what we were doing. We've looked at a revision lesson so far on meiosis. We were looking at the processes, we would try to identify the different phases. We just looked at some questions where we tried to identify different structures, the parts, and obviously characteristics of the different phases. In this segment, we're going to look at what happens when meiosis goes wrong. We're going to probably try and look at some questions that are based on how you would write an essay on meiosis, and then probably we will have some time to be able to look at when meiosis goes wrong and how do we identify those characteristics that we see in a karyotype. So let's get straight into the lesson and try and unpack what happens when meiosis goes wrong. So guys, we often refer to mutations taking place, and we know that there are mutations that take place at a DNA level. So we refer to those as gene mutations. We also need to recognize that sometimes mutations happen at a chromosome level. And this is often when meiosis doesn't occur correctly. And so we find that might affect the chromosome structure. It could affect the number of chromosomes in the gametes. So let's try and understand what is the impact of these chromosomal mutations that have on a cell. So there's an important concept that we need to recognize as non-disjunction. And essentially non-disjunction, the word disjunction refers to separation. The concept of non-disjunction again refers to when separation does not occur. So it's important that we recognize that separation normally or disjunction of chromosomes occur in anaphase 1 as well as in anaphase 2, where the chromatids move apart. Now, we find that in certain chromosomes that this does not occur always, and that can create a non-separation of those chromosomes. So, non-disjunction occurs when homologous chromosomes fail to separate during anaphase 1 of meiosis, or the sister chromatids fail to separate out during anaphase 2. This will then result in one of the gametes having 2 copies of one chromosome and the other having no copy of that chromosome. The other chromosomes are distributed normally except for the pair that have not separated. We refer to this as chromosome non-disjunction. If either of these gametes then unite with another gamete during fertilization, this will result in a cell that is aneuploid, or having an abnormal number of chromosomes. So if I were to illustrate that using a human gamete, so we know that during the normal process of fertilization, we have 23 from the sperm, 23 from the egg that come together and give you a complement of 46. However, we do recognize that sometimes when non-disjunction occurs, we have a cell that either has 22 chromosomes or 24 chromosomes. And when these fertilize with other cells that are normal or having a full haploid set of chromosomes, they will result in 23 plus another cell having 23, will give you a total of 40, if I do that correctly, 45. And that's a cell that has an abnormal number of chromosomes. It's one less than the full 46. Likewise, if this gamete that has an abnormal number of chromosomes fertilizes with a gamete that is having a full haploid set, it will result in a cell that now ends up with 47 chromosomes. And having 47 chromosomes is one more than the full diploid set. And what we need to recognize is that when individual cells have an extra set of chromosomes, there's a significant impact that it has on the individual, mainly because a chromosome has many genes, and so that gene expression is either compromised or overexpressed, depending on the type of aneuploidy. And we will look at that in a bit. Right, so this is an overview of what happens during non-disjunction, again, the non-separation. During meiosis 1, we see that the normal process is when chromosomes move to the opposite poles during anaphase 1. However, during non-disjunction, we find that towards the one side, there is no separation of chromosomes, and we see the entire pair being pulled to the one cell. If this cell then was to fertilize, you'll end up with a cell having an extra copy of that chromosome, and that means it's an abnormal number of chromosomes. We also know that if this cell that has not received the chromosome fertilizes, there would be a chromosome missing for that chromosome in that cell. We also know that during meiosis 2, there is the separation of the chromatids during anaphase 2. Here we see that sometimes when that does not occur, the entire chromosome is pulled to the one side of the cell. And this results in this daughter cell having 2 copies of that specific chromosome. The other cell that did not receive the chromatid ends up with having a chromatid that's missing. And so again, the scenario of one cell having one more and the other cell having one less. And so we find that this might also occur in mitosis, where the entire chromosome is pulled to the one side, resulting in 2 sets of the one chromosome in a cell. And that's essentially showing you how non-disjunction happens and what that leads to. We'll discuss that in the next bit. So we say that when when cell division or meiosis goes wrong, we have what we refer to as one example, which is Down syndrome. And Down syndrome, again, is called 21 trisomy. 21 trisomy referring to an extra set of chromosome number 21. And the usual number of chromosomes that we have for each is a pair. Unfortunately, during a Down syndrome fertilization, we have 3 copies of chromosome number 21. Okay, people with Down syndromes have a total of 47 chromosomes, and this approximately affects 1 in 700 children and alters their child's phenotype either moderately or severely. And so we see different effects of this abnormality, and we see sometimes phenotypically lots of characteristics that are identified with these individuals, and we'll discuss some of those in a bit. It's important that we understand what is a karyotype and how karyotypes are used to recognize abnormalities. Essentially, a karyotype is an image that is created of a cell's chromosome arrangement. And so we find that it illustrates and arranges chromosomes in pairs, and those pairs are arranged according to their size and shape from the biggest to the smallest. We also know that karyotypes, in this case, help us to identify the gender of an individual. And clearly we can see here, this is an individual that has an X and a Y, and that makes this the karyotype of a male individual. What is highlighted in this little box is the presence of 1, 2, and 3 chromosomes number 21. So this initially is a sign of an abnormality in the chromosome number. That means that this individual has an extra copy, a trisomy of 21, and this is typically seen in individuals that have Down syndrome. Right, so let's move on to a possible essay question on meiosis. It's important that we recognize again that essays can be set on a question in meiosis where we'd have to discuss various different processes or even when meiosis goes wrong. So let's look at one topic and let's see how to answer that question. So describe the structure and arrangement of chromosomes making up a normal human karyotype. So guys, a normal human karyotype is essentially an individual that is not affected by any genetic disorder. Also describe the behavior of the chromosomes during the different phases of meiosis 1. So if we look at this, there are two parts to that essay. Remember that there's content mark as well as the synthesis mark. It's often a question that is rushed towards the end, and when you're rushing a question and if you do not synthesize the essay, you're going to lose marks here. Remember that an essay is not a flow diagram, it is not a bullet point. We want to see facts that are well synthesized and linked together. So let's look at what the planning of this essay looks like. So when unpacking the question, we recognize that there are two parts to the question. The first part requires you to describe the structure and arrangement of chromosomes in a karyotype. That's the first part. The second part of the question requires that we describe the behavior of the chromosomes during the different phases of meiosis. So guys, remember that the essay is 17 marks. So you've got to now kind of figure out what are the number of facts that I need to give for each process. So I generally would rather give 8 there or probably 9 facts there and give another 9 here so that I have a collective of 18 facts. And that will take me all the way up till 17 at least. So think of what you need to do before you actually start writing. So I'm going to think of 9 facts for that part of the essay and another 9 facts for that. Let's see if that then can get us the full 17 marks for this. So as I mentioned, we've got to describe the structure and arrangement of chromosomes. The examiner has allocated a maximum of 8 marks. So yes, by giving 9 facts, at least we will get the minimum 8 that are required. Okay, so again, the structure and arrangement of chromosomes are important. Remember to discuss that chromosomes at the start consist of chromatids, which are held together by centromeres. Reference that a human karyotype has 23 pairs of chromosomes, and these are arranged according to their size, they're similar in lengths, they have genes that are the same location, meaning they code for the same characteristics, and that these have alleles at a particular location, which we refer to as gene loci, and that they have the same centromere position. And if we go back to our karyotypes, one would recognize that it's the position of the centromere that defines the shape of these chromosomes. And that is what geneticists will use when arranging chromosomes in a karyotype. We also need to recognize that each somatic cell has 22 pairs, of which there are autosomes, and there are a pair of gonosomes, which are the X and Y chromosomes. We know that autosomes are arranged in pairs from the largest to the smallest. We need to talk about the males having an XY chromosome, which determines the gender of the males, as well as the females having 2 X chromosomes. We also need to recognize that the X chromosome is a larger chromosome and the Y chromosome being significantly smaller. The other part of the essay requires us to discuss the behavior of the chromosomes during meiosis 1. So again, it means that you've got to go back and discuss prophase 1, metaphase 1, anaphase 1, and telophase 1. It's important that you recognize the key descriptions to prophase 1 is the significance of crossing over, the concept of how chromatids change information at the point called the chiasmata. During metaphase 2, we need to recognize that this is when chromosomes arrange themselves along the equator. And during anaphase, how these chromosomes move apart. And then finally, in telophase 1, where we discuss how 2 daughter cells are produced, and each of these cells have half the original number of chromosomes. And again, guys, we often tend to see that learners forget about the 3 marks, which I often use as I use this rubric to explain the significance of those 3 marks. So it's called the synthesis, and the first mark is for relevance. So this information is about being relevant to what you're talking, and this, in the context of this it was about the human karyotype. It was about the different phases of meiosis that will give you the 1 mark. In terms of the logical sequencing of your information, it's important that you're able to logically lay out your information in terms of the sequence of events that happen in meiosis 1. And then finally, in terms of how comprehensive you were, in terms of the number of facts for each part of the question. Guys, here's an indication of a karyotype that you must be familiar with and being able to identify. Guys, we've reached the end of our lesson. It's amazing how quick we've gone through different components of meiosis. The fact that we've looked at different characteristics and understood that, and we're able to use that in understanding how to unpack that in an essay is fundamental. We've also looked at what happens when meiosis goes wrong. I trust that you've enjoyed the lesson, that you've been able to make some key notes. Please go back, look at your textbooks, try and attempt a few questions, look at the answers, and I trust that you will then improve your understanding on this section. Guys, you've been a fantastic audience. Wish you well. See you sometime soon. Have a bye-lekker day.